Well, I guess we've given a couple minutes. So maybe I'll get started and introduce you. So it's certainly our pleasure to welcome you today, Dr. Zhong. Um, I mean, Dr. Wang. So Dr. Zhong Wang is a career computational biologist at Lawrence Berkeley Lab and program head for the Genome R&D analysis group, I guess, at the DOE Joint Genome Institute, which I take is in California. He received his BS in microbiology from Shandong University in China in 1994, his PhD in cell biology from Duke in 2004, then completed a postdoc at the Institute of Genome Science and Policy at Duke as well, and became a research scientist at Yale in 2008, and also director of the bioinformatics group at Yale Stem Cell Center. His research included metagenomics algorithm development, scalable software solutions for genomic analysis, and AI applications in genomics. Dr. Wang has published over 40 papers, including papers in Science and Nature, and he's co-authored a book entitled Introduction to Computational Metagenomics. So quite a lot of accomplishments, and we'd love to hear about what you have to talk about today. And again, if we want to do this informally, that is wonderful for us. And so just welcome, and I'll let you take it from here.
Thank you, Dr. McKenty, for the very nice introduction. It's always a pleasure to talk to Dr. Fang, because I've known Dr. Fang for a long time. So when he proposed for me to share some latest research with the folks here, I accepted more than happily. So so let me share my screen. Can everybody see my slide?
Yes, we can see it.
Okay, let's do this. Slideshow. Enters. All right. Dr. McKenty just give me talk about my kind of professional life. So I like to give you more or less a personal version for that. So I was born in a farmer's family in eastern China, a city called Qingdao. So the city is actually known for its beer. I'm not sure if you like this light lager type beer. So I was very interested in nature. Growing up in a farm. Then went to college in Shandong University and studied microbiology. I thought microbes are really powerful and cool. And then I got really intrigued in the molecular genetics of microbes. So I went ahead to study molecular biology and cell biology in Chinese Academy of Sciences in Shanghai. Spent 3 years there. Then I joined Duke. Again, study stem cell, the genetic program of stem cell, and then did a postdoc there. More or less understand stem cell biology from the aspect of genome organization, how the protein organized to express genes. Then after finished my postdoc, I went to Yale, and I'm really attracted to new technology, especially this next generation sequencing. So they are working on RNA-seq technology. Then I was recruited in 2009 to California in the Joint Genome Institute, which is part of the Lawrence Berkeley National Lab. I mostly focus on scalable data analysis, software development for metagenome, which actually we'll spend some time talking about this today. I also like to mention that I'm also affiliated with a University of California at Merced, the youngest campus among the UC system. I really enjoy, my hat as an educator there. So I want to start with metagenome. So for this audience, I'm not quite sure, is this concept new, to you? But at least you probably have already very familiar with microbiome. We know, as a person, every cell we have, there are 10 microbes for it, cohabitat with us. I always introduced the concept of microbiome to my computer science colleagues. Say, hey, if you guys have like a 10 seconds intimate kiss with your loved one, you guys end up exchanging 80 million of bacteria. So these microbes, they're actually everywhere. They're not only associated with human and other animals, they're also associated with plants. There's in all kinds of terrestrial locations. They're also in water, in the air. So we study them, the metagenomics. We ask basically 3 types of questions. Who are there? It's a diversity question. We want to survey the different kinds of microbes in this environment. And we're also trying to understand what they're doing there. So more like a functional metagenomics perspective. And also, we're curious about how do these microbes interact with each other, and also how they respond to the environmental changes. So this is the last question is more relevant to JGI, because we are funded by the Department of Energy, and study how microbes respond to climate change is one of the key research areas we do. So by the way, I really like the presentation to be conversational. So if you have any question, feel free to stop me. Turn on your camera, and so I can see you too. Okay. So we study metagenomics. We end up generating a lot of data. That's actually one of my main jobs at the Joint Institute, to develop software for dealing with these large data sets. So the community really generates many large data sets. So just to name a few, you guys are probably well familiar with the Human Microbiome Project. So these are a large collaborative project with for this single project. There's over 30,000 samples, generate over 40 terabytes of sequences over the years. At DOE, we study the soil, the NEO, which is agricultural soil, and also the Great Redox experiment, that's the Harvard Forest soil. And in both of the projects, generates 3 to 5 terabytes of sequences. We also study freshwater microbial communities in this Lake Mendota. There's over 10 terabytes of sequence were generated. The largest environmental measurement project is probably the Tara Ocean. So this is a global survey of this microbiome in oceans across the world that generated 35 terabytes already. So analyzing this data set isn't easy. So in the past 15 years or so, my group colleagues at the Berkeley lab and also from the community as well, we were able to leverage the supercomputers the Department of Energy built. Like here, I showed the picture of Frontier. That's the fastest supercomputer in the world. And we also leverage the cloud computing like AWS and Google Cloud. So we are part of, or someone from my group too, we develop several tools like the MetaHimer. This is a terabyte scale metagenome assembler. It can assemble trillions of short reads into large genome fragments. And my group also developed a MetaBat as a binding tools is to further group this a large fragment into draft genomes. And people predict millions of billions of genes and proteins from these data sets. So how we study them, as first step, you can actually cluster this genome in these proteins or genes into the clusters. Then you can select the representative to study. So this HIP MCL is able to scale up to really large data. And we also recently developed a framework called Axolotl that's based on cloud computing technologies, which give you more flexibility. Because all these assembler binders, they can only do one job, but data analysis often requires flexible, ask specific question the tool may not be able to address. So I will not talk about these tools today. I just want to quickly showcase how capable these tools are. So this is one example. I had a collaboration with Dr. Morgan Case at Miami University, and also Feng Chen from Illumina. So in this project, from a single sample in underneath the thick ice in Antarctica, we sequenced the microbes, generate 16 terabytes of data. So with this amount of data, we were able to assemble them in less than 1 hour with 1,000 nodes on Frontier. Similarly, this group have put together the entire human microbiome projects. The data size is almost 100 terabytes. Get 23 gigabytes of assembled of all the human microbiome project. So the most complex microbial communities from this Mendota dataset. From this dataset, we actually put together 105 gigabytes of assembled metagenome, and with 160 million genes predicted. So these tools really give we give a lot of big catalogs. You've probably seen lots of publications on Nature, Cell. So the key question I was asking now we have so much data, we predict so many genes, and how much insight have we learned from this huge amount of data. So this this ChatGPT moment is a very good inspiration. Similarly, like in the natural language area, over years we have accumulated so much text, right? In Wikipedia, in Twitter, and the large language models can actually learn the representation of human language, and they can generate artificial text, and they can talk to us as if they were human. So today, I actually want to ask the question of, is, for genomics, for metagenomics, are we also entering a ChatGPT era? So the analogy here is, can we train large language model based on a large, diverse metagenomic data set? I just showed you some of those, and learn the representations of genomic sequences, and hopefully generate artificial genome sequences as we wish. So to do that, a lot of these large language models will start from a step called pre-training. So in this step, we basically try to teach the model basics, every topic in genomics in this case, and then we can use this called the foundational model to do feed them with specialized data set so that it can do special things. So to train a function model, we started with a diverse set of this high quality metagenome data set. It really covered, ocean, forest, plant associated, and also including this data set that we just put together, and also have 52 gigabases of sampled human microbiome too. And together, we collected 700 gigabases of assembled metagenome. So these are not finished reference genome. The reason we decided to use metagenome instead of the reference genome we have from NCBI is because we thought the reference collection is probably a biased representation of all the microbiome life there. So we only use the sequence data alone, nothing else. We try not to use any human generated knowledge. So the question is, can the large language model learn the language of genome or the knowledge of genome just from the sequence alone? So unlike ChatGPT, the word, the vocabulary concept is pretty clear. For genome, from the time we actually don't know if genome is or a language, what vocabulary it would be. So people in the field, like a group of people from Argonne National Lab, they developed a genome model called GeneSlam, which actually won the 2022 the Bell award. So they use codon, which actually makes sense, right? Because codon is the unit for genome to encode protein, but it's probably has some restriction because there's a lot of non-coding region or regular elements. Not quite sure codon can represent those elements. So to overcome these limitation, group people from Stanford, they developed another model called EVO. They just use single letters, AGCT, to have the flexibility to represent the genome. We and our collaborators at Northwestern, they actually developed a DNA BERD model. They use those frequent words, or in this case, cameras, could be variable length. So this really has nothing to do with biology at all. It's basically a mathematical representation of the sequence for efficiency, because we're trying to use the smallest collection, the smallest dictionary to represent as much sequence as possible. So we trained a model, we named it Genome. It's based on the popular transformer architecture called Mistral. We had to do several ticks to make it run fast. We use these mathematical BP tokens with about 4,000 words in the dictionary. It's a fairly deep architecture with 24 layers, and we end up with a 4 billion model. Since this the first time we do it, we actually don't know whether this size is big enough. And nevertheless, that's how it trained. And because this size of model comfortably fit into our computational budget, so the whole training cost 15 days on the supercomputer, the NERSC Perlmutter supercomputer. That's the supercomputer collocated at the Berkeley lab. So this is done on 64 NVIDIA A100 GPUs. So with this model, the model has basic 3 functions. It can generate sequences up to 50 KB. Also, given a sequence, it can output embedding and representation. I will talk about that a little bit later. And also, given a sequence, it can compute its likelihood of, I assume, how likely this sequence is drawn from the training data. So because a lot of tricks we did, so the model is actually incredibly fast compared to the other 2 genome large language models for embedding. Genemotion is 10 to 20 times more efficient than the other 2. And for generation, it's even faster. It's 1,000 times faster. For biologist colleague, the fastest DNA polymers that generates DNA in nature is about only 1 KB per second, but Genemotion is 10 times faster. Can generate 10 on a single node. It can generate 10 KB per second. But interacting with the model turned out not to be that easy. So I'm not sure how many people have seen the movie Arrival. So when I got the model, so this is, the exact feel, I have. So basically, you have to interact with the model in the format of DNA, and we'll have to think hard what is mean when it gives us something. So for the rest part of the talk, so I'm going to share my experience interact with the model. I have to warn you though, a lot of the time we actually don't know what the model actually means. We can only guess. So the first thing, obviously, does the model understand the genome language? So here I just want to show one example. So the model can actually speak fluently in DNA. It generates sequences containing full-length enzymes. Here, it's a piece of DNA I submitted to BLAST, and BLAST shows, it contains 2 genes, and both of them have specific hits to known proteins in the database. If you look at this protein alignment, if I didn't know this is generated by the Genemotion, I cannot tell this actually any different from like a natural protein we found in metagenome. So I jokingly tell people that the model kind of passed the Turing test in biology. Turing test is a test Alan Turing proposed in AI field that AI can fool human, or they behave like human, then they have general intelligence. So then this is another example. So in this case, the model made a transmembrane protein apparently because it contains a non-cytoplasmic domain and a cytoplasmic domain separated by multiple transmembrane domains. In this case, it's 6. So if there are 6 transmembrane domain, actually we'll put the 2 ends, the 2 terminate of the protein on the same side of the membrane. So I call this model as adept in biology, but probably terrible in math. But sometimes it does get it right. Like this time we had 2 transmembrane domain, the 2 sides on the same side of the membrane. So here's another example. So we know that ChatGPT can autocomplete of the human natural language synthesis. So we asked the question, this model can autocomplete proteins. So we basically gave the model, it's a 400, or I don't remember. Yeah, it's a 400 amino acid protein. We basically say, okay, here's 200, the first 200. Can you complete? And the model complete it without any problem. So actually, here we try to align. So the blue one is the machine generated, and the orange one, the gold color, is the natural counterpart. So the protein structure is predicted by EM fold, and you can't really tell the difference of the two. If you look at those protein alignments, so the first part, are the prompt, they're identical, and you look at the generated sequence, they look highly similar to as expected. So is this autocomplete, does the model simply memorize, maybe the protein is already in the print set. So we did one experiment. So in one experiment, we systematically mutate the DNA sequence. You remember Genemotion is a DNA model. It operates at the DNA level. It does not know anything about the protein because we didn't tell anything about it. So in this experiment, we systematically mutate the DNA sequence by switching the codons so that the protein sequence hasn't changed, because these are synonymous mutations. So with 10%, 10%, 30% of the codons are switched, model is still able to complete the protein without a problem. Only see a slight drop when 40% of the codon are replaced. In a control experiment, if we introduce these non-synonymous mutations that actually change the protein, so we give model like a wrong prompts, and with 10%, 20%, 30%, the ability to complete the protein is almost completely abolished. So from this kind of experiment, it appears that the model understands the meaning of the prompts. Meaning means, here is protein coding. So again, I want to stress on it's not straightforward to figure out how the model does this. So if you, the model can really do this pretty consistently. It's not like like a typing monkey, a monkey with a typewriter, just random type of things. So here I look at another protein, and also can autocomplete, generate very good protein structure to a natural one. So here I have about 30 natural version of this protein align each other, and it shows a very conserved site shown red here. So these are from one experiment. We also generated 30 of these artificial proteins, and you see the model knows where to be conservative. And again, this is even remarkable because model use a vocabulary. We actually purposely make it harder, right? Because it's not even codon. So nevertheless, it knows where to be conservative. So we, with this, we could spend really forever to test the model, how it works, but probably the model still we actually don't know what it cannot do and what it does do well, and because our evidence, our experiment down on the model is still very limited. But I think the key question here, can we actually use it? Realizing that the model is likely to be wrong. For this audience, probably you're already familiar with the famous quote from George Box. All models are wrong, but some are useful. So the question, can we use it to do something useful? So one thing we try to do, because I work with metagenome binding, we always try to group pieces of DNA from the same genome together. In the field, people are using this tetranucleotide frequency. It's kind of a very simple representation of the sequences. It basically works. You take DNA sequences, you count the frequency of all the formers, and then you transform a sequence into a vector, and then you can do dimensional reduction into 2-dimension, and hopefully different pieces, each dot of the same color is actually a DNA sequence from the same species. So the TNF is pretty useful to group in this simple community with 10 genomes together. You can see it does fairly well on many of the genome, but some of the genomes, they may share some of the fragments, so these genomes are close to each other. Some genomes are really close, like E. coli and Salmonella. It's very hard to discriminate these two using TNF. So we did the same thing with genome ocean embedding, and compared to the other two models. Compared to that two, the genome ocean works really well for this community, and it's even outperform TNF. So so the other thing we can do, we can generate for smaller data set, metagenome data set, we can actually generate more data. So in this case, we start with a few DNA fragments from a metagenome community. This shows in blue. We're trying to generate more data with the large language model. So the gold color is the genome ocean, and green is AVO, and red is GenSlim. So we generate more data, then we use a fourth model called a nucleotide transformer to be the judge to see which model generates more diversity. So because this reflect the generation, whether the model can generate this species level diversity, and it's pretty clear that genome ocean does a fairly good job, much better than the other two. When we're looking about functional diversity, so we generate thousands of contigs. As I said, we can generate a lot of data pretty quickly. Like a typical metagenome data set, 2/3 of the genes actually are hypothetical or unannotated. About 1/3 can be assigned to known functions. So among these 1/3 generate sequences, they actually spread across a fairly large spectrum of different functions. Here's just one example. It's a long contig, long fragment contain multiple gene annotations. I just to show this because the model actually can generate backward. See, all the genes is actually going from right to left, but the generation direction is from left to right. So I think this is probably because in the training data, the information is encoded actually on both strands. So another thing we are just start to explore is whether we can find these functional elements in the genome. So we know that there are quite a few protein large language models. So some unique advantage of genome large language model is actually you can study these functional elements like promoters, transcription factors, binding sites, splicing, epigenetic marks. So in this case, we simply scan a piece of DNA, systematically mutate the bases in the prompt, and then see its effect on the generation. And this way you can see these cis interactions. So here you just a yeast arginine tRNA gene, and then the red hot array turned out to be the mature tRNA. And we guessed, the reason the model generates signal is probably because they're like base pairs, because of this segment structure forming in mature tRNA. So we're still in the process of playing with the model and see what else it can do. So before I switch to the drug discovery part, are there any questions from the audience for the part I just talked? Okay. Okay, maybe you guys are all waiting for this interesting part, right? Okay, drug discovery, because I know talking to a medical community, I think this topic might be more interesting. So I just showed I stole a figure from a recent review by George Church's group at Harvard. So this review, they systematically reviewed the three stages in drug discovery. So from understanding the disease mechanism, drug discovery, and then finally to clinical trial. So in the past, yeah, by the way, this is like $2 billion segment. So it may grow into $2 billion in the next 10 to 15 years, I think. So in the past, we normally just manually review the literature, do patent search, and then combine with the slow experimental laborious natural product discovery process, and then subject this natural product to random screening. And if we're lucky, we will find a match, and then we can go to do the manual patient trial matching. And the current technology have greatly facilitated this process, with Google, with all these powerful search tools, and combined with functional genomics, multi-omics data set, and analysis tools, we're able to conduct a virtual screening. But still, the structure-based drug design, although with AlphaFold, with this recent Nobel laureates work, this process has greatly improved. But still, there's a lot of manual process in it. And of course, for the clinical trial, it's still largely manual. So in the future, this author proposed there will be a lot of these robotic or agent type powered by large language models, from automatic target gene identification, biochemical, pharmaceutical principle discovery, to design those therapeutic modalities, automatic experiment for validation, de novo molecular generation, instead of relying on natural product. This also LLM can also be a crucial part for clinical trial, like automatic patient trial matching, trial design, and result prediction. So for I've talked about this genome function model. So I think, in this process, at least in some of the stuff like this de novo molecule generation, we may be able to help to this vision. So, but how? So for people not familiar with natural product discovery, so a lot of natural product, at least in bacteria and fungi, they are encoded by these biosynthetic gene clusters. So you have an array of genes right next to each other on the genome. They include core and additional biosynthetic genes. And sometimes they have transporter and regulatory genes and some other genes. These cluster ranges from several kb to hundreds of kb. These, you can think about these are assembly lines. They take a subject and sequentially add more groups to become a bioactive product. So they're experimentally have determined quite a few, but still probably just a few hundreds. And then combining with bioinformatic tools based on homology-based and also rule-based, there are about a few thousand well-annotated PGCs in the database. We actually took this and searched against all sequence reference genomes and recovered about 12 million of these potential predictive PGCs. And we're in the process of mining all the metagenomes we have. And we estimate there are at least 10 times or over 100 millions of PGCs out there. But if you look at this natural product being approved by FDA that's actually become drugs, there are actually very few. So here, like at least 10 of those approved drugs. So how could we greatly speed up this process? So the idea, okay, let's train a large language model. So I mentioned that we train this genome ocean functional model. By itself, it does not generate biosynthetic gene clusters, although it has some basic understanding of the genome in general. So to educate the model, we feed the 12 million PGCs we have from the JGI's secondary metabolites collection group. And then we end up another foundation model. We just call it BGC-FM. We're hoping that it can generate novel green metabolites, or in this context, natural product with therapeutic capabilities. And we also, from a genome biologist perspective, we can help this model hopefully improve the existing BGC annotations. So this we recently started this process. We don't have a lot of data to share yet, but I just want to show this slide. So this is a 40 kb DNA fragment generated by this BGC foundation model. So we use the state-of-art tools called anti-smash to predict, to look for new BGCs. And anti-smash again give really high score for this find. Thanks, this is T1 PKS. It contains all the core biosynthesis genes. And if you look at it closer, not only it has all the right component of the genes, but also within each gene, it has the correct array of modules that can support a complete production. So this is, I don't think there's any other model that is capable of understanding this high order organization of those genes, because you your model need to understand not only meaning of each individual genes, right, to have the right domain architecture, but also you need to have the right combination, of these genes. So we're collaborating with the synthetic biology group because they are actually manually come up with these combinations to find new chemicals. So hopefully, our model can help them to design new drugs. So I think I'm going to end here to leave a few minutes for this quiet audience. So I show example of training a specific large language model. I believe there are many scientific domains could benefit from similar research. I show that we use diverse data, efficient architecture, combined with a lot of tests to test this model. We're going to open this model to the research community for clinical genome biologists, for functional annotation, find disease causes, therapeutic development, making artificial proteins and biochemicals. And if you're already trying to develop a smart agent to help your research, this model could be a bedrock for your agent. There are quite some challenges. I will not go through them. These, I think the biggest challenge is how you interact with this model, because the model does not speak in natural language yet. So we're actually in the process of working with collaborators, try to add an adapter so we can actually directly talk to this model. There's a certain risk. I think the biggest risk is probably because we cannot reliably tell the generated sequences from these natural ones. And imagine that these sequences got leaked to NCBI, because everybody rely on the database for their research. This might have a pretty bad consequence if they got submitted there. This, BGCs, a lot of BGCs actually encode toxin. People may just use it to design to poison people. I don't know. Probably not that easy. So I want to so where are our way in terms of AI in science, in genomics? So I think clearly, we're already way past the stage that this a monkey with a typewriter, because it takes like the of universe to type out a Hamlet of the Shakespeare's play. It's probably better than the same dogs. It turned out, this is something new. This is people finding this new Guinea dog can actually sing a few songs. Not quite sure if the dogs understand the song, but we're probably still far away, at least in my opinion, to the age of her that we have this smart agent just understand what we need, give exactly what we want, and respond to the demand for our research. So I will probably put Genomotion here. So this research is really a lot of collaboration between researchers from the Berkeley lab at JGI and Northwestern. So I want to special thank Dr. Han Liu's group at Northwestern. He has talented students help us to train the models, and former students. And now she's also faculty at Northwestern, so undergrad, help us to evaluate these models. And a lot of the data actually provided by Georgia Institute, especially the IMG group, and also the secondary metabolite collection group. And it's also benefit from very talented summer students. She's actually an undergrad from John Hopkins. Yeah, sure. She's still working with us on the BGC research. And the computation is supported by the National Energy Research Scientific Computing Center. It's benefit from the supercomputers there. So thank you for your attention. Are there any questions?
Well, first off, thank you, Dr. Wang. And I think, David Alexander, you're on there, right? I'm not sure that everybody has the ability to unmute themselves. Is that true, David?
Yes, they can unmute. They can come off mute. They can turn on their
Okay.
camera, and they can join in. Yes.
Okay, great. I wasn't sure about that. Okay, great. So we'll welcome it. Any questions then?
I don't have a question, but just a comment. You talked about the infinite monkey, and I kind of thought about that at the same time. But then another analogy came to mind, and that's Hitchhiker's Guide to the Galaxy, where they spent years building the deep thought to answer the question about the life, the universe, and everything. And then when they got their answer 7.5 million years later, they couldn't understand it, so they had to build another computer. So I think you need another computer to solve this.
I love that analogy. Okay, so maybe yeah, I will add that to my slide. Yeah, so I mean I think these large model things really make us think, right? Now with the ChatGPT's 01, large models seem to be able to think as well, right? A lot of my colleagues here, I mean, we have a lot like debate. I mean, very friendly debate, because we not quite sure how these large models work. I mean, do they truly have intelligence? Do they truly understand language of genome, or they simply memorize things, right? But if they memorize things so well, they know where their attention should be, like in all the conserved domains or the important sites. But shouldn't we call that learning, right? And they can paraphrase using their own words about life. Shouldn't we call that learning, right? So I mean, either way we cannot prove it, right? Because the model, it's very hard for the model to join our conversation yet. But I'm very keen to talk to people, share this model, and ask questions. What can the model can do for you for your research? I think that's a lot of my colleagues in this field are focused on, right? Because we could spend like the rest of our life to figure out how this model work, because it's a black box. I don't know if a lot of people try to develop this white box approach. I don't know how successful they are, but a lot of people think actually that's probably the wrong direction, right? We probably just focus on, hey, how could we use it? I mean, ChatGPT is probably very wrong. It's probably just the wrong model, but it's incredibly useful. I mean, we use like 80% of my code already produces by ChatGPT. Yeah.
Our own thought process are very much black boxes as well. I mean, half the time we don't know why we're doing anything. So, I wouldn't worry too much about it.
Exactly. Yeah.
Any other comments or questions from our speaker?
Who want to use this model? Raise your hand.
Yeah. It's definitely a black box to me.
So we are collaborating with NVIDIA. So hopefully, because not everybody has, supercomputers to to do this. We're working with them to make a cloud version of the model, so everybody can just use it, without worry about setting up the environment, run the inference. Yeah, so stay tuned if you want to use it.
Great. Well, thank you again so much for taking time out and sharing your research with us. Very exciting stuff.
Okay. Thank you. Take care.
Bye-bye. You too.